ring around the rosies was based off the plague that devastated all of Europe. The rosy rash is a symptom of the plague. The posies of herbs used to be carried to ward off the smell of the disease, and ashes, meaning the blackened skin of plague victims, as well as the burning of their homes after their death. So far, there have been 3 major pandemics of the plague. The causes of the pandemics was the bacterium Yersinia pestis, which is spread through the bites of flea and rodents. Symptoms include unusually high fever, swelling of the lymph nodes, which causes a bubo, a swollen, inflamed lymph node in the armpit or groin. The bacterium Yersinia pestis is a deadly disease if left without proper medical treatment, allowing it to be a great contender for bioterrorism. There are 3 variations of the plague to consider for bioterrorism: bubonic, pneumonic, and septicemic. The bubonic and septicemic plague occur via infection of flea and rodent bites. The pathogen enters the lymph nodes, where it grows to high numbers, thus causing a bubo. Once in the blood, the pathogen is considered septicemic, and the spleen and liver begin to try to remove the bacteria. Yersinia pestis is victorious because it is able to avoid and restrain host cells' immune reaction. The pathogen can resist phagocytosis by injecting proteins called Yersinia outer proteins into macrophages and other immune cells. These injected proteins form pores which through other Yersinia outer proteins travel to get to the cell cytoplasm and limit phagocytosis. As the organisms grow within the blood, liver, and spleen, it spreads throughout the body, similar to other bacterial infections. Pneumonic plague occurs from the inhalation of infected droplets. It can be passed on from human to human without the bite of fleas or rodents. This is the most dangerous form of the Black Plague because it is so easily passed on, thereby resulting in a high fatality rate.
Yersinia pestis is classified under a chemoorganotroph, which uses aerobic and anaerobic processes to produce and consume hydrogen and carbon dioxide for energy. Yersinia pestis uses binary fission to reproduce. This is a process in which a cell splits in half and produces an identical cell. The process of growth occurs at temperatures from 4 to 40 degrees Celsius. However, optimal growth is from 28 to 30 degrees Celsius, with a pH ranging from 7.2 to 7.6, extremes ranging from 5 to 9.6. Humans and rodents' body temperatures sit around 37 degrees Celsius, making them perfect hosts for the bacteria. Yersinia pestis is a rod-shaped gram-negative bacteria that corresponds to the Enterobacteriaceae family. It is a prokaryotic cell containing a cell wall, cell membrane, capsule, ribosomes, nucleoid region, flagella, and pili. The cell wall is a lipid composition and an enterobacterial antigen, making it easy for the cell to attack immune cells. Yersinia pestis primarily comes from the Yersinia pseudotuberculosis. Yersinia pseudotuberculosis is the least common of the 3 main Yersinia bacteria. Yersinia pestis is a highly uniform clone of the Yersinia pseudotuberculosis, which has evolved throughout the years.
The first main pandemic and first occurrence of the Black Plague was the Plague of Justinian. This outbreak erupted in an Egyptian port city during the summer of 541 AD. The disease quickly spread out to the countryside and over to Europe and North Africa. All of the lands bordering the Mediterranean were affected. The second pandemic of the Black Plague started in India, China, Russia, and Western Europe. It hit again in the autumn of 1347. In less than 3 years, it had hit almost all of Europe and areas of Greenland as well. The third pandemic of the Black Plague started in the mid-19th century and slowly made its way through the Yunnan Peninsula. This last pandemic eventually reached Hong Kong in 1894. From Hong Kong, a steamship carried the disease across to many coastal cities around the world. Unlike the first and second pandemics, the spread of the disease was limited to coastal cities. Instead of killing millions like the first 2 pandemics, it killed only about 100. The Black Plague has nastily been used in biological warfare. One major place the Black Plague was used was at the Siege of Caffa. It was thought that the plague was spread by Europeans hurling diseased cadavers over to Caffa, causing the Italians to flee. The bacteria could have been transmitted to living humans when they are disposing of infected bodies. The bacteria often traveled through cuts or wounds of those disposing of the bodies. It is estimated that a very large amount of cadavers were used, increasing the transmission of the disease. Because there were so many victims of the disease, there were many bodies to be disposed of, and therefore many bodies were being hurled over to Kappa. For many years, treatment of the Black Plague was very poor. It was based on mysticism and superstition. The sick were given mixtures of blood as well as tablets made from rattlesnake meat. Several years later, the victims were given purgatives, which are laxatives, diaphoretics to simulate sweating, and emetics, an agent to promote vomiting. In the late 1800s, the Yersinia pestis bacteria was isolated, in which scientists tried to develop a cure. Some scientists discovered the use of antiseptics like iodine were helpful with some patients. Currently, victims are being treated with antibiotics. Antibiotics were suggested by the World Health Organization Expert Committee in 1970. There are several different antibiotics patients are given, such as doxycycline and tetracycline. Some Yersinia pestis becomes resistant to these antibiotics, so others such as streptomycin and kanamycin are used. Thankfully, after years of research and experiments, scientists have been able to find a more effective treatment than superstition and mysticism. Thankfully, the Black Plague is not found very much in America because we have much more sanitary conditions as well as very good health care to treat any plagued by this disease. Another reason the Black Plague is not as common in America is because most individuals show a moderate resistance to infection. This resistance results from previous exposure so that nearly all individuals have antibodies against plague antigens. They may also carry an inherent characteristic of the species. These two things cause mild signs and a low mortality rate.